Hello, good evening everyone. Am I audible? Is my voice clear? Yes. So, good evening everyone. As you know, most of you are know me, and some of you are regularly following the sessions. My name is Ubaid Tariq, and I am taking this problem solving session of this course named Cell Culture Technologies every Tuesday at 7 p.m. So I welcome you all in this problem solving session. So today we are solving the 7th week assignment of this course, and we will directly start with the questions. And as you are aware that this is the 8-week course, and maybe next week will be our last session. So we will directly start with the questions. So first question, which of these can be used to separate population of two cell types from a mix? Four answers are given. A is immunopanning, and B part is fluorescence assisted cell sorting. C is both A and B, and D none of the above. Anyone? Yes. Some of you have given the correct answers. Can anyone from the audience tell me why do we need to separate the cells from the mixture of cells? There are two questions from my side. Why do we need to separate the cells from the mixture of cells, and in which case we will get the mixture of cells? You can directly unmute yourself, or you can write in the chat box. So I think no one is answering. So for example, you may have heard about the pure culture or single cell culture. In the single cell culture, if we need to study the effect of some particular drug on a specific population or specific cell type, so in that case you need a single cell in a cell culture. And usually in the cell lines, the cell line is already designed by some agency, and they have designed them, and they have given when you procure these cells, you will get the single cells. But when you isolate the cells by yourself from the organ, from the tissue, in case of the primary cultures, you directly isolate the cells from the tissue. For example, in case of heart, if you take the heart tissue and disintegrate the extracellular matrix using collagenase and other proteases, so you will get all the cell types in the culture media which are present in the heart. And usually mostly in case of heart, two most common cell types in heart are fibroblasts and cardiomyocytes. So when you grow the cells, so in the cell culture you will get both cardiomyocytes and fibroblasts also. If you want to work on the fibroblasts only, so there should be some technique to only get these fibroblasts only for the further culture. Or if someone needs cardiomyocytes, some techniques are there which so that you will get only one cell type from the mixture of cells. And that's why these cell techniques are used. These are the techniques which are used, but there are also some simple based on the cell type. There are also some simple techniques which are used. For example, in some case, in some organ there are two cell types. one cell type adheres quickly to the cell culture surface, and another cell type they adhere very later, maybe 5 to 6 hours later from the first cell type. So in that case, we know that the cell A, it adheres quickly. When the cell A adheres, we will give the washing and remove the cell B. That is the simple technique, and it only works in few cell cases. But mostly sophisticated, these high-end instruments are used, like fax and immunopanning is used to separate the cells. So I will discuss more about this fax fluorescence assisted cell sorting. So what happens in this fax? For example, you have the mixture of cells. The cells, you have the different types of cells, and these cells are suspended in a sheath fluid or some fluid or PBS or other fluids that are used in this fax. And when you suspend them, and what this fax do, it aspirates these samples, and after aspiration there are two types of detectors present in the in this fax. So in the fax you can do two things. One is the you will be able to know the cell, which type of cell is this, and you will be also able to separate the cell at the same time. For example, it has two types of detectors. One is FSC and the SSC. So this FSC detector, it detects the size of the cell. For example, if you have the two types of cells having the different size, it will detect on the basis of size. So healthy or dead cells also have the different size. It can also detect between the healthy and dead cells. And this SSC detector, it detects the fluorescence or granularity. So on the base of size you can also detect cells, but another detector it detects the cells on the basis of fluorescent labeling. So you know that every cell has some particular type of markers, particular type of antigens that are present that are specifically present in some type of cell, like T cell and B cell. So there are specific markers. So you what you will do, you will attach the antibody to that specific markers, and that antibody is also that antibody is also labeled with some fluorescent tag, and that fluorescent tag will help to detect the different types of cells. So so these detectors help to detect on the basis of size, or on the basis of granularity, or on the basis of fluorescent labeling that's present on a specific type of cell. And these cells here also it is shown that like is the cell, and there is a antibody which has a fluorescent dye attached to it, so we will be able to detect it. So for example, this green cell and these blue, they are two types of cells, and you want to isolate them in a separate these ependophs, or separate these tubes. So what this fluorescence assisted cell sorting do, as these detectors have detected that this and this cell is a different, so they will in this tube different charge will be given to these cells, and different charge actually will not be directly given to the cell, but there will be some sheath fluid around this cell, and the charge will be given to this sheath fluid, and below this tunnel there are deflection plates. For example, positive charge is given to this blue type of cell, and every blue type of cell now onwards this positive charge will be given, and to the green cells this negative charge will be given. And because of this deflection, these cells after coming out from this tunnel or and this nozzle, they will be separated in these tubes. So we will be able to separate and first detect the cells, because in the cell mixture you will not be able to visualize what type of cells. First the detector have to detect which type of cell is this, and then they will be separated. If there are some other cells or some debris that will be present in your sample, that will not get any charge because they will not be detected by these detectors, and it will be collected here. In between these there is a waste bin or waste collector. It will be collected in here is another tube. So this was all about FACS. And another cell sorting techniques or cell these isolation techniques are immunopanning. So immunopanning is also the technique in which the cells they have the, as you know, as I already said, that the separation is only possible because each cell has some specific markers. So because of that specific markers, the antibodies are attached to the cell. So the antibodies will help in the separation of these cells. For example, here if you see here in this this is a tube, there are different types of cells present, and if we want to separate this type of cell having this this color, this brownish color, so this we know that it has some specific marker, so we will design the antibody that is specific to this marker, and this antibody gets attached to this and this cell only, and after that we will put it in another plate, and that plate has already some antibody labeled attached on the surface, and that antibody has an affinity of binding to this antibody. So if we give the washing, this antibody, this pinkish antibody, and our cells will be adhered, and we will be able to wash other cells. So our cells will remain adhered to this surface of the plate. We will remove other cells, and later we will use some solvents to remove our cells also. And when we take the supernatant from this cell, for example here in this fluid, the washing, we can, if we need to separate the cells also here which are in the washing, so we can take these washings and place in the other this tube or this plate, and this plate has a different types of antibodies which are specific to other types of cells. For example, this grayish or bluish type cell, this one. So this bluish type cell was not adhered in this plate, but it will get adhered to this plate. So now in the second step, this cell will be isolated, and simultaneously we can do it for different number of cells. So this is the plate type, this immunopanning. Another is the bead type immunopanning or magnetic immunopanning. But it has recently different companies have used this magnetic field to separate the cells, and it is trending nowadays. So what is done in this? So you have in this tube you have the sample, and the sample is here, like it is shown that 4 to 5 type of cells are here, but you want to but your desired cells are only this orange type of cells. If you want to separate these orange type of cells only, or these blue type of cells, these orange ones are the magnetic beads, actually micro or nano size magnetic beads. So these gray or bluish type are our cells that we need to separate, and these cells will have the specific markers, and we will design the antibody specific to this marker, and this antibody will be attached to this cell. But this antibody has also this magnetic bead, nanomagnetic bead or nanomagnetic particle attached to it. So when first we will mix these antibodies which are attached to these magnetic nano beads. After that we will pass them in from this tube, and this tube also has a magnetic field attached here. So because of this magnetic field, and as these antibodies, they are attached to the cells, as they have the nano magnetic particles, they will remain adherent to this magnetic, because of the magnetic force of attraction. So they will remain there. Other cells will pass from this tube, and they will be collected in the bottom. Later on, when the magnetic field is removed, so these cells will be separated, and you can collect these cells separately. So these were the techniques which are mostly used in to separate the cells. There are other simple techniques. Can anyone from the audience tell which they may be you may be using them in your labs? There are some other common techniques also, like centrifugation. If the cells have different sizes, so using the centrifugation also at different speeds for different types of cells, different types of cells can be separated. But those are not specific techniques, but these like FACS and immunopanning, they are very specific to some particular type of cell. So moving to the question number 2, which of these is the signature of all leukocyte groups? These here are given some markers like CD45 positive, CD45 negative, CD340, CD340 negative. So these are some markers which are given, and which of these markers is present in the all types of leukocytes? Someone has asked question maybe from the previous question, why use this most use centrifugation? Can you rewrite your question? If I am able to understand it, you are saying why are we using these immunopanning, these techniques to immunopanning or FACS as compared to centrifugation? Is this your question? Okay, why not centrifugation? Yes, as I already said, centrifugation is the non-specific technique, and these immunopanning or FACS, they are very specific techniques. So if we want to, for example, in case of, first I will explain about centrifugation. Centrifugation will only isolate cells or separate cells on the basis of density, and it is pretty much it is less possible. So the cells have the different densities. Mostly the cells have the same density, same size, but in some types of cells, if there is a significant difference in the size of the cells between the two cells, so you can easily separate them by using centrifugation at different speeds. But that is also not the specific technique. You may get, for example, if you are using some particular type of speed, for example 1500 rpm you are using for a particular type of cell, but some other cells may also be may pilot down at this speed. You are not pretty sure. If your experiment is such that, so it is not specific to some particular type of cell, you only want to separate cells. If there are some other cells also, you have the, in your experiment there are, if you have this chances that if there is some error that it is okay, so you can use centrifugation also. But in some sophisticated experiments where you only want to separate some particular type of cells, so in that case only techniques which are focusing on the specific type of markers of the cell. For example, every cell has a specific marker, and now here there are no chances that other cells will also be separated. If you see in this case, like this magnetic thing, so here these cells have some antigens or some markers that are present, and these markers are only present on this type of cells. So this antibody and this magnetic bead, they will only attach to this type of cells, and only this type of cell will be separate. So it is more specific. So that's why these immunopanning or FACS, they are used mostly in case of cell sorting. Is it clear, Selvam? Thank you. So question number 2, which of these is the signature of all leukocyte groups? So A part is CD45 positive. These are Selvum. These are like I was saying about markers. These are some markers. These are some antigens which are present in the cells. So leukocytes have some specific markers. There are different types of leukocytes, and there is a one common marker in all types of leukocytes. There are also other markers also which are different in different types of leukocytes. So the answer is CD45 is a common marker in all these leukocytes. So here I have shown in this slide. So all the leukocytes have these CD45 marker. So these leukocytes, as you know, that they are derived from the myeloid progenitor cell. So there are different types of these cells like granulocytes, monocytes, T lymphocytes, B lymphocytes, these thrombocytes. They all have this common marker of CD45. So CD45 is present in all of them. So if we use the antibody, CD45 antibody, to separate these cells, so we will get all these types of cells. So if we want to separate all the leukocytes, we will use CD45 antibody, which is specific to CD45. So they will attach to this CD45, and by the help of magnet or by the help of FACS using fluorescence tags, we can separate all the type of leukocytes. But for example, in some experiment, you only want to separate monocytes. So in that case, you will use the marker that is specific to monocytes. So there are some specific markers of every leukocyte. For example, monocyte is CD14 marker. So you will use the antibody which is specific to this CD14, and this only binds to the CD14, and you will be only to isolate or separate these monocytes. And same is the case with others, T lymphocytes, B lymphocytes, thrombocytes, granulocytes, all have some specific markers also. So it depends on your experiment. If you want to only isolate all these leukocytes, so you can use the common marker which is present in all the monocyte, all these leukocytes, and you will design the antibody for all these. So you will be able to separate in all of them. But if you want to, if your experiment is on some specific, so you will use the specific markers and you will design the specific antibody. Here in this table, I have also shown what other type of markers are present. Like CD45 is the common CD4 positive, and T cell receptor, TCR, B positive. So there are some types of markers. So here in case of T regulatory cell, there are FOXP3 positive marker, D25 marker. So these are all the markers of these cells. And every type of cell in our body, they have some specific markers. So you can separate, or you can isolate, or you can study any type of cell using these markers. So question number 3, this is a very simple question. And dash is a protein molecule that recognizes dash on a cell surface. So there are 4 answers. Antigen is a protein molecule and recognizes antibody on a surface. Second answer is antibody is a protein molecule and recognizes antigen on a surface. Third is the antigen, and fourth is the antibody. So what is the answer? So like I was saying in earlier these questions, I was explaining about the same thing. Antibody is a protein molecule that recognizes the antigen on the surface. So antigen is present on the surface of the cell. So that antigen is recognized by the antibody, and so this antibody binds to this antigen. So B is the answer. So here in this slide, I have shown that antigen and antibody. So this this is the antibody Y-shaped. It has 2 chains. This is the heavy chain, this is the light chain. So 2 heavy chains are present and 2 light chains are present. And this one here, this portion is called constant region, and this portion is called variable region, and actually this portion is called a variable region. And this, why this is called variable region? Because this is variable. There is actually VDJ recombination. There is a recombination between the some parts of these antibody or these proteins. So it will by the help of these variables, you will different types of antibodies, different types of these. Actually, they are the receptor types, and these receptors here they are called paratops. Actually, it's not a receptor; it's the binding site. So these binding sites on the antibody, they are called paratops, and because of these variable regions, variable region in the heavy chain, variable region in the light chain, and because of the recombination that happens here. So large, if we have the thousands of antigens present in our body, so because of these recombination, the thousands antibodies with the thousands of these different sequences here in the paratop, they are formed in our body. So in the antigen, there is also some protein on which this antibody binds, and that site is called epitope. So the antigen has an epitope on it is a site where the antibody binds, and the antibody has also the site in the variable region, which is called a paratope, and this paratope binds to the epitope. And if this paratope recognizes the epitope, if they are same, so this epitope will bind to here if there is a binding site here. Like you have studied about this, like the lock and key model. So here in this picture, in the left side, I have shown that these antibodies, they are specific to the antigens. So if you have one antigen A, so for the antigen A, there will be the specific antibody which only binds to the antigen A, and any other antibody will not bind to this antigen A. So there is a specificity in this. So interaction only happens when their sites match. So here I have there are some examples, like here this is a cell, and this cell has different types of antigens. This, for example, this is antigen 1, and this triangle type is antigen 2. So so there in our body there will or if you want to design the antibody for this cell, so for the antigen 1 there will be some specific antibody. So antibody here is given antibody 1. So antibody, if this specific antibody is provided in this medium, this antibody 1 will bind to the antigen 1. So antigen antibody reaction occurs, and it will bind to this. So here is antigen 2. So antigen 2, for the antigen 2 specific type of antibody is there, and this antibody only binds to the antigen 2, it will not bind to the antigen 1. So antigen antibody interaction only occurs in this case when the antigenic sites, and when this paratope in the antibody and epitope in the antigen, when they match. So here in this antibody 1, it will not be able to bind to antigen 1 because it is not specific to this, and this antibody 2, it will not be able to bind to antigen 1. How to find antigen specific type or common? So your question is how to find the antigen. Okay, it's clear. So it is known that, for example, it is a discovery. For example, in different types of cells, they have already, like I was talking about leukocytes, it is discovered that they have different types of antigens present on the cell surface. Some are common to all these cells, and the sequence of these antigens are also known, and the antibody which binds to these antigens, it is also known. So if you want to study some particular type of cell, and you know the antigens present on it, the antibody is also known, you have to order the antibody from the vendor, and you can use them in your experiments. So this specificity is here in the paratop. This paratop is in when actually this antibody is formed. It will be the lengthy lecture. If I will go in this direction. When this antibody is formed, at the time of formation of this antibody, this antigen, this epitope of the antigen, it is shown to this antibody. When it is shown to at one step, which is called I don't know exactly the name of that step. If I remember that, I will let you know. And this epitope is shown to that antibody when this antibody is formed. So this antibody now it is made for this specific epitope also, and it knows this epitope. So it will bind to only this type of epitope or this type of antigen. So that's why they are specific to this. So we can design antibody for any type of antigen. So when they are formed from the B cells, so there are some steps in the there is some molecular mechanism how these antibodies are formed, and during the formation of these antibodies, these epitopes, they are shown to this antibody in the cell. So here the paratope, the sequence of the paratope is such that it will only bind to this antigen, to this epitope only. So that's why they are specific, and they will not bind to any type of antigen, whatever. If there is even 0.1% difference in between the two antigens, they will specifically recognize the only antigen for which they are made in the cell, from the B cells. So another question. In DASH, there are two types of motor neurons. One are in DASH, there are two types of motor neurons. There are two types of motor neurons. One are small in size, and other are large in size. So what is the answer? So answers are they are the parts of spinal cord, dorsal horn, ventral horn. They are the parts of brain actually. Dorsal horn, ventral horn, dorsal horn, and ventral horn. And in A part there are alpha and gamma. Anyone knows the answer? So the answer is ventral horn, so the answer is D part. So in the ventral horn there are two types of motor neurons. One are small in size, that is called gamma neuron, and other is large in size, which is called alpha neuron. So here in this picture I will explain this. So here is the dorsal horn or dorsal root, and here is the ventral horn. So from these ventral horn, alpha and gamma neurons, they come from these motor neurons, and they go to the effector organ. So here why they are called large, and why, like here it is shown that. So from this ventral horn, so this is the ventral horn from this right side and left side. So from this type of right ventral horn, one gamma motor neuron it will come out from this ventral horn, and one alpha motor neuron, and from this left side only one alpha motor neuron will come out, and it will go to the spindle fibers or other muscle fibers. So they are the motor neurons. Motor neurons give the information from the brain to the effector organ while the function needs to be done. So question number 5 is, in right spinal cord at E14 only motor neurons express a particular marker. This information can be used to. So the question is, in the right spinal cord there is some location E14, there are some motor neurons, and these motor neurons, they express a particular type of marker. So every so these markers, from these markers, which type of information can be collected? So so the answers are, these markers can be used to isolate these cells from the mixed culture with other cells of the spinal cord, test the electrical activity of these cells, digest these cells, none of the above. So what is the answer? So you should be able to answer, as I have explained these things already in the earlier questions. Yes, Sandhya. Answer is A. So as I already said, because of these specific type of markers that are present in different types of cells. So neurons, they also has a, if there are some specific markers, and there are present some specific markers are known for the neural cells or different types of neural cells. And because of these neural cells, when we from the rat, when we take the spinal cord and when we digest it, so different types of cells, glial cells also, neurons also, they will come in the media. So if we want to separate some isolate some specific types of neural cells, so these markers will be helpful. So A part is the answer. So because of this particular type of marker, we will be able to isolate these cells from the mixed cell culture, mixed cell culture of the spinal cord. When, as you are taking the spinal cord as a primary culture from these from the spinal cord, you are taking the cells as a primary culture, so you will be able to isolate these or separate some particular type of cell using the specific marker that are present in different types of cell. So another question is question number 6. Trypsin is used to dash by designing the extracellular by digesting the extracellular matrix as it is so it is a very basic question. Anyone of, as you have, this is the 7th week of the cell culture technologies, and most of you must be knowing that what actually trypsin does. What is the function of trypsin? So the answers are, trypsin lifts cells off the surface, and it is a condensation enzyme. B part is lift culture cells off surface, it is a proteolytic enzyme. C part is attach cell culture to the surface, a condensation enzyme. Third part, D part is attach the cultured cells of the surface, a proteolytic enzyme. So what is the answer? Most of you have answered this question. So this is the proteolytic enzyme, and it lifts culture cells off the surface because it digests the extracellular matrix. So here I have shown, so why we need to lift the cells from the surface. Anyone? Adherent cells, they are attached to the bottom of the flask, so they will only, as they are anchorage dependent, they will only grow attached to the bottom of the surface. So if we want to passage the cells, or we want to detach the cells for any experimentation like FACS or some other experiments, or simply we want to passage the cells to grow them in a large number of flasks, at that time, as the cells are attached to this flask, so you will not be able, when you remove the media, cells will remain there as they are attached to the flask. So why these cells will be attached? When you seed the cells, after some time, as they are anchorage dependent, they will settle in the bottom of the flask, and after that they will excrete extracellular matrix. as in the normal physiology, they are embedded in extracellular matrix. Here also in the flask they will secrete the extracellular matrix ECM, and because of that ECM they will remain attached to the surface of this flask. So to remove the cells, you have to use the proteolytic enzyme which digests the extracellular matrix, or which detaches this which disintegrates the extracellular matrix or ECM. This, because of the digestion of its ECM, the cells will be detached from the surface. So what is the process? First you will remove the media from this flask, and the cells will be in the surface. Then you will give the washing with the PBS, so that the remnants of the media and FBS, they will be removed. After that you will use the trypsin. So trypsin EDTA is usually used. So this trypsin is added to this surface, and after that this flask is incubated for 2 to 3 minutes depending on the type of cell. It is incubated at 37 degrees centigrade in the incubator, and after some time when you see these cells in a microscope, you will see they will be detached from they will get detached from this surface. So then you will add the media to the flask, or you will or you can transfer them into 2 or 3 flasks to passage them, or to split them, or if you want to do some other experiments. So there is also you have to follow this procedure. So after adding the media to this flask, so here why we need to add the media? As the trypsin is a proteolytic enzyme, that means it disintegrates or digests proteases. First it will disintegrate ECM, but if you keep the cells for more time in this extracellular ECM, in this trypsin can also damage the cell or this cell membrane, because it's a proteolytic enzyme. So you have to dilute it after some time. When cells get detached, you have to dilute it with the media, and after that, as this trypsin is a proteolytic enzyme, this should not be present in the media after the passaging. So we have to remove this media. How we can remove this media? Use containing this trypsin. So we will transfer them in a falcon, and this falcon will be centrifuged at some 300 G, or here in labs we are using, I think 300 G, we are using 1000 RPM we are using. So we will get the cell plate, and this media will be removed, and this cell plate can be resuspended in a fresh media, and now it does not contain any trypsin, and now it is resuspended, and it is transferred to the other flasks for the growth. So this is the actual protocol when you passage the cells or split cells. Which one of these is not a component of B27 supplement? So answers are vitamin A, biotin, acutase, and sodium selenite. So I have attached the answer here, so you can directly see from the slide only. So can anyone tell me what is B27 supplement? Why it is used? Anyone? So B27 supplement, it is usually used for the neural cells. Earlier, firstly it was used for the culture of hippocampal cells, as these neurons have some specific requirements for their growth, and this B27 supplement provides these components for the effective growth of hippocampal neurons, or all other types of neurons. First it was used for hippocampal neurons. So the answer is C. As you can see, this acutase, it is the answer. As you can see, these are the components of B27 media. saying this vitamin A is present, also biotin is also present, this sodium selenite is also present, this acutase is not present. So the answer is C. Glutamax is dash and has dash then glutamine. So glutamax is L-alanine L-glutamine, and it has a lower stability. There are 4 answers given. What is the correct answer? Selvam, you have raised the hand. Have you any question? Okay. So this glutamax is L-alanine L-glutamine, and it has a higher stability than glutamine. And in the answer I have explanation for this. I have written here. You can also read that. So L-glutamine it is the essential nutrient in the cell culture. It spontaneously this L-glutamine, it is an important component of this cultural media. And actually this L-glutamine in the cell culture, it is spontaneously or it is abruptly degraded in the cell culture media. So after degradation, it generates the ammonia and pyrrolidine carbonic acid as a byproducts, which are also toxic to the cells. So this company have made its alternative or its derivative, which is glutamax. So glutamax is this L-alanine L-glutamine. It has a modification. Alanyl group is also attached to this. So it is a L-alanine L-glutamine. Why it is made? As the L-glutamine has a low stability. So because of adding this L-alanine group to this L-glutamine, so it becomes highly stable. So it has higher stability as compared to glutamine. So this glutamax supplement is an improved cell culture supplement that can be used as a direct substitute of L-glutamine in the cell culture. So this was all about this question. So the answer is B part, L-alanine L-glutamine, and this has higher stability than glutamine. So question number 9 is dash is a supplement used for serum-free fetal hippocampal neuron culture media. So I've already given the answer in the earlier question. Which of these supplements are used for the growth of fetal hippocampal neurons in a cell culture? So yes, Sandhya. B27, as I already said, B27 is a specific type of media or supplement that is used to grow the cells to grow the neurons or hippocampal cells in the cell culture media. So these statements they are related to the animal origin free media. So here all of these statements are correct. So the answer is 4, A, B, C, D. So all of these statements are correct from for this animal origin free medium. So when you order your samples, when you order your media, so if this is written animal origin free media, or in some cases if you specifically need the animal origin free media or medium for your cell culture. So you should remember all of these things. They are these things are in this animal origin free media. So all of these statements are correct, and I will read these statements for you. So it is not manufactured using any raw material ingredients derived from an animal, including human tissue, cell or body fluid. So animal origin free, as the name suggests, that it does not contain any raw material or any ingredient that's derived from the animal. That is why it's called animal origin free. And animal humans also come, or different tissues or cells or body fluids also come. So in animal origin free medium, all these ingredients or raw materials from any type of animal tissues or cells or body fluids, it should not be present. That is at the primary level of manufacturing. So in the primary level of manufacturing also, these animal materials or animal raw materials for the synthesis of this should also not be present for if it is an animal origin free medium. No animal derived materials are used as starting material or in the primary level manufacturing process. So these animal ingredients should not be directly present in the medium also, and also no derivatives or no raw materials should be used from the animal source for the manufacturing of any component of these animal origin free medium. So it should be completely animal free, animal component free. And another statement is, the components used to manufacture the raw material ingredients are not derived from an animal tissue, cell or body fluid at the secondary level of manufacturing. In the secondary level of manufacturing also, they should not be animal tissue derived or cell derived. What this secondary level of manufacturing means, for example, component D is present in this cell culture medium, animal origin free cell culture medium. So this component D, it is made from component B. So that component B is made from component 1. So the component 1 should also not be from the animal origin. So if the component 1 is from component A is from animal origin, so from the component A, component B is made, from the component B, component D is made. So if the component A is from the animal origin, so it is not animal origin free. So at the secondary level of manufacturing, this the derivatives or raw materials should not be from the animal, cell or body fluid origin. Another statement is, ingredients and the components may be produced by or extracted from plants, bacteria or yeast, or cell-free systems derived from them, or may be produced by organic synthesis. So as the animal components are not present, so what ingredients or components can be present in this? So ingredients and components may be produced from or produced or extracted from plants, bacteria, yeast. So if the components are produced from plants, bacteria and yeast, or any cell-free systems, so that they can be included in this animal origin free medium. Or if the components are produced by some organic synthesis, in a chemistry lab, if they synthesize some components using using different chemicals, and by this organic synthesis, these all components can be used in the animal origin free medium. And last statement is, these animal origin free medium may contain recombinant animal or non-animal proteins, provided they are expressed in plants or microorganisms. For example, there are some essential components which are only present in animals, like amino acids, some growth factors. They are only present in animals, and if they are very essential for the growth medium, for the growth of cells, and In animal origin free media, they can be added, but they can be produced by the recombinant DNA technology. So the animal proteins, if they are necessary to be added, so they may be isolated as from the recombinant DNA technology. But that using that recombinant DNA technology, if the protein A is necessary, and protein A is an animal protein that should be added in this animal origin free medium, that protein, for the recombinant DNA technology, we should know the gene of that protein A, and that protein A, gene of that protein A should be expressed only in the plant cell or animal cell, or plant or animal. And after expressing in the plant and animal, there this animal protein will be formed using RDT, but it will be formed in the plant and animal, and after that it can be used in this animal origin free medium. So these are all for the statements. You should remember all these four. So this was all about this session 7. So thank you for joining the session. So this is my YouTube channel. You can subscribe to this channel, or you can have a look at this channel. I will upload all the earlier videos, and now onward all the videos will be uploaded here. And there are also other videos you can see that if you have missed something in the lecture. So thank you so much for joining this session. If you have any other questions, maybe related to today's session, or maybe you have other questions, you can ask in the chat box, or you can directly unmute yourself. So Cell Vem has asked that how to grow the cancer cells. So cancer cells are like other cells. They are the normal type of cells. Only the difference is, there are some checkpoints in the cell culture, cell division control. There are some checkpoints when the cell grow. There are some checkpoints which will control the division of these cells. So that control is not present in these cancerous cells. So they will grow. They will grow, and the control is not present. So if there is like in the normal cell, if there is a DNA damage, if the cell will grow in the cell will not pass the checkpoint, and cell will not grow. But the cancerous cell, they pass the checkpoints and they will grow, uncontrolled growth. So in the cell culture, we can normally grow them in the cell culture like other cells. They will like the other cells, they will attach. The cancer cells, actually, they are also cancer cells of these tumors adherent. If the normal cell is adherent, the cancer cell will be same adherent. But their growth is not controlled, and maybe they will express some other markers also, because of as they are cancerous in nature. So in the cell culture, we grow them like other cells, normal cells. Any other questions? Thank you, Selvam, for interacting. I expect everyone should interact and ask the questions, so the session will be good. It will be interactive. So if there are no questions, I will conclude this session, and I will meet you in next coming week at 7 p.m. So thank you so much for joining. I will conclude this session now.